Sacral neuromodulation is currently used to treat worst-case patients who suffer from fecal and urinary continence and incontinence. Current modulation methods require a neurostimulator and a lead to be surgically implanted within the patient's sacrum. This invasive therapy can cost up to $46,000 and comes with multiple risks. Efforts to stimulate the sacral nerve at the S3 foramina non-invasively have been unsuccessful due to a variety of problems. Stimulating from the skin surface to the depth that is required will cause damage, burning, and pain to the patient. Higher frequencies can be used to increase depth penetration. However, nerve stimulation at high frequencies is shown to be ineffective. This poses the idea of using the superposition of two high-frequency electric fields with a slight difference in frequency. Current literature demonstrates the feasibility of the stimulation method. Animal studies show that non-invasive deep brain stimulation on the hippocampus of a mouse can reach the motor cortex without stimulating any overlying cortex. Computational models show stimulating the sacral nerve in people is also feasible, and benchtop trials have been attempted. However, due to a lack in electrode technology, all benchtop trials are conducted using 2D electrode arrays. My project documents the design and production of a 3D electrode array to determine the power distribution of resulting electric field packets in benchtop trials. 25 plunge electrodes, a PCB, and a stabilizing plate are the key design features of the 3D electrode array. 25 typical 16-gauge needles are converted into plunge electrodes, as a single traditional plunge electrode can cost up to $1,000. Each needle is cut identically using a custom-made needle alignment jig. A second jig is then used to evenly space 0.2 mm holes 5 mm apart in a heat-treat tube. Wires are then threaded into the evenly spaced holes, and the needle is then inserted into the mask. Finally, an epoxy is injected into the plunge electrode mold and removed after 24 hours. An alignment plate is designed to evenly space the electrodes 10 mm apart. The plunge electrodes are then sandwiched between the alignment plate and a PCB that connects the electrodes to an acquisition device. The 3D electrodes are then tested using a benchtop phantom and chicken breast. The biosemi was connected to the array, and the transformer channel isolated 10 stimulator was used to stimulate both the phantom and the chicken breast models. Analysis of the center electrode from benchtop trials show expected results of a 20 Hz envelope. A FFT analysis of all the channels is used to show the amplitude of the electric fields. The variations in amplitude are likely due to the location of the electric field packet and the locations of the electrodes. For stimulation using identical electric fields, it is expected that there should be no electric field packet. This can be seen by looking at the results of same frequency stimulation and the FFT analysis. For future workings, a 4D visualization method is required to depict the X, Y, Z and amplitude of the data. This is to accurately visualize the power distribution in both the phantom and chicken breast models. Special thanks to my supervisors Saeed and Melanie and the research team at Alighaleh. Ko te aroa te waka, ko tarawera te maunga, ko wharengaro whakarangatira tangata tōku ingoa. Mauri ora.